Solamente hay 2 niños. Hello. Hello, guys. Hello.
Hi, Miss.
Hi, good morning. Okay, guys. Today the class is the science, okay? The professor is Elias. Okay, Elias. Go, please.
Hello. Hello.
Hi.
Students, how are you all? Hmm. Will you allow me to share the screen? Roxana.
Sorry, it's ready.
Okay.
Yeah, it's good.
Okay. So today our topic of discussion is about the scientific method. What is actually a scientific method? You all may have gone through some problem in your life and you tackle all this with some perceptions and with some ideas. So to deal a problem with the specific ideas and observations and a terminology is logically called as a scientific method. So what is a science? Science is a systemized knowledge that wait a minute. Okay. Science is a systemized knowledge derived from the observations and experiment. Uh, Science is systemized mean it is a process of some occurrence in our thoughts that we may observe in our problems, and all we do what we have to solve the problem with some techniques that are our experiments. So science is basically a systemized knowledge that is derived from the observations and the experiments. These experiments are carried out to be determine the principle about how nature operates. There are a lot of examples about the science and the scientific knowledge we will discuss in further details. Here is what is biological method. A biological method is a scientific method in which biological problems are solved. So all the biologists solve the biological terms and techniques with the help of a scientific method. It comprises the steps a biologist adopt in order to solve a biological problems. There are some steps that are to be kept in mind when you solve the scientific method. What is actually a scientific method? It is a logical and organized way of solving of solving a problem. So first of all, we have to define problems and observations. Then we move toward the collect background informations, and then we move to hypothesis, and then we test hypothesis. Then we make records and observation. At the end, we draw conclusions about the problems and observations we have made. So first of all, we will see what our observations are. Observations are made with the 5 senses that are visions, hearing, smell, taste, and touch. These are the 5 things that are used to make observations for all the problems that we have to deal with. So there are 2 types of observations. The one is qualitative, while the other is quantitative. The qualitative observations, see the name quality. This will show us about the quality of observations. For example, here is an example about the freezing and boiling point of water. Both freezing point and boiling point are the properties of single substance, which is water. So we say that freezing point of water is colder or lower than the boiling point. The boiling point of water is 100 while the freezing point is 0. So here is only we discussing about the quality of water. It may be a freezing or it may be a boiling water. So here we are getting the idea about the quality of water. And maybe another example, a liter of water is heavier than the liter of ethanol. Here are the two substances. The one is water while the other is ethanol. So here what we are discussing, the one is about the substance mass, that is in our liter form, while the other also in the form of liter form. So what we are getting from it, we say that a liter of water is heavier than the liter of ethanol. So the properties of this substance telling us about the quality of both two substances, water and ethanol. What is quantitative observations? In these terms, we get the some quantity, some numerical value of the substance. So here is a freezing point of water is 0 Celsius and the boiling point is 100 Celsius. What we are getting from this term, we are getting the freezing point is about 0 Celsius while the boiling point is 100 Celsius. So we are getting a numerical value about the single substance. So it's telling us the quantity of observations. In the second example, we are getting about the substance mass, that is a liter of water weight 1000 gram and a liter of ethanol weight 789. Here we are comparing the masses of two substances. Okay, so there are two types of observation. One is qualitative while the other is quantitative. So which one is better, qualitative and quantitative? The only accurate and precise observation is that is quantitative, because we are getting about the numerical observations or numerical values of the substance, so we can make more and accurate data about the problem solving. The next step is defining a problem. What is defining a problem and collecting the background information? So when we have a problem, we go to the background informations, from where it comes and how we have to deal with. So identify the problem is a major issue in the biological or scientific method. So here we can have an example about the acid rain. So acid rain is a phenomena that destroy the buildings, skins, and other etc. So here is the problem. What are the effects of acid rain on the salamander? We are discussing the specific species salamanders. So this is the problem with a single species. Okay, so we are identifying a problem. The problem is the effect of acid rains, while the other time is collecting informations, that is our background informations, that how we do, that is we should know the normal development of salamander as well as the characteristics of area that are affected by the acid rain. That this means the areas is affected by the smog, or maybe affected by the more fire, or maybe the pollutions in the air that may cause the acid rain in the environment. So these all are the informations about the acid rain that we are getting from the data that we have collected for the problem solving. So the next is formulating a hypothesis. The first step we do is the observation, and then we do the hypothesis. What are hypotheses are? Comparing and contrasting different types of observation. Good morning, guys. Okay. So the next step we do after getting the observations about a simple problem, we do a hypothesis. What our hypotheses are actually? It is a simple guess or maybe an idea that we have made about the observation to solve that. Okay, to solve the problems, we made a guess about the problem that may be an educated way of solving the problem. It is a proposed answer to the question or problems. Maybe if the problem is simply about the disease, we may have a questions about the disease, where from it comes and how it affects the people. These all are questions, and to answer these all questions, we formulate a hypothesis. So basically, hypothesis is a proposed answer that answer to the all questions that originate by the observations. It is a statement that can be tested. After proposing a question answer session about the observations, we do a test on the hypothesis. What hypothesis is basically by definition. So its definition is the tentative explanation of observation is called a hypothesis. What does tentative mean? Tentative mean it is a collective or maybe subjunctive idea that have collected from all the observation that we have made from the questions and the answers about the problem. It may be defined as a proposition that might be true. It not always be true, but sometimes it may be true or it may be false. There are some characteristics of hypothesis. It should be or it may be a general statement. Okay, it should be a general statement, or it should be a tentative idea. It should agree with that available observation. All the observation or question answer we have made about the problem, it must be agree with all the observation we have made with the problem. So it should be kept as simple as possible. It should not be complex or perplexed with the observations, so we can solve the problem easily. Okay, it should be kept as simple as possible, and it should be testable and potentially falsifiable. Means the hypothesis we made from the observations must be testable. It can be performed in the lab and on the species, or might be on the field that we are applying on it. And it potentially falsifiable means it not always be true. It can be falsified by the experiment after doing experiment. In other words, there should be a way to show the hypothesis is false, a way to disprove the hypothesis. The next step after doing hypothesis is deduction. What deduction is mean? Deduction are the logical consequences of hypothesis. These are the result of hypothesis that are deducted from the deduction mean deducted. These are carried from the hypothesis. So for this purpose, a hypothesis is taken as a true and expected result. This process is proceed if the hypothesis is true. Otherwise, the hypothesis we made, we have to check again, and we have to make another observations about the problem. So for this purpose, the logical consequences that have been made deducted from the hypothesis is taken as a true and expected result or draw from it. What are the expected result for the problems? If we are dealing with the problems of the malaria or maybe some disease, there might be some thoughts or maybe some ideas in our mind that we can concluded from the hypothesis that we have made in our mind. So we can draw the deduction about the problem. This involves the use of if and then logic. Always start with the if and end with the then. So this is basically a hit and trial method to solve the problem. From start to deduction, we cannot say that we proceeding a process might be a true or not, because after deduction we have to do a experiment on the problem that show us if the process is going to be proved by the experiment that will support our observations. So then we proceed next. Otherwise we have to move for backward for the more observations and the logical answers for the question answer session. The next is types of deductions. What are the two types of deductions are? The one is deductive reasoning, while the other is inductive reasoning. In the deductive reasoning, we move from general to specific. Here is one of the examples. If we accept check out this is starting from the if. So if we say that if we accept all birds have wings, then sparrow are birds, then we conclude that, and it is going with the then if and then trial methods. So if we accept all birds have wings and sparrow are birds, then we conclude that sparrow have wings. This is a simple method. We are moving from the general to specific. What is general? We all know that all the birds have wings. This is our general thoughts about some species. So if we moving from the general to specific, specific means a single species that we are considering, that is a sparrow. So if we accept that all birds have wings and the sparrow are birds, then the sparrow have wings. In the inductive reasoning, we move from specific to general. Specific means we forward with the we have to go with the general or specific species, and then we conclude conclude our reasoning with the general thoughts. If here is an example: if sparrow have wings and are birds, and we know eagles, hawks, parrots all are birds, then all the birds have wings. This is a general statement, all birds have wings, while if we say sparrow have wings, this is a specific specifically termed sparrow. It is a single species. We are not considering the parrot, hawks and eagle. So only we are discussing about the sparrow. So this is a single species, that is a specific, and we conclude all birds that are eagles, hawks and parrots, that is a general statement about a single phylum of the birds. So if sparrow have wings and are birds, and we know eagle, hawks, parrots are birds, then we induced all birds have wings. These are the two types of deduction. The one is deductive reasoning, while the other is inductive reasoning. After making a reasoning about the hypothesis, we proceed with the testing a hypothesis. So if our observation and that hypothesis are going to be true, we do a test. An experiment is a controlled procedure designed to test a hypothesis. Testing the hypothesis also means the performance of the experiment. So, so testing these hypotheses, there are two methods to test the experiment. The one is controlled and the other is experimental method. In controlled experiment, we provide some basic, we deal with the single terms that we have to make about the problems. For example, we say we discuss about the photosynthesis. So there is a single factor carbon dioxide. If we have two jars about in which one is with the light while the other one is in the dark, so we can make a conclusion about the photosynthesis that and we know that the photosynthesis require light to proceed. So the jar with the light will proceed and make a glucose, while the jars in the dark will not produce the glucose. So an experiment is a controlled procedure designed to test a hypothesis. It is an in an experiment, one variable or a condition is changed and the response of another variable is measured. In testing the hypothesis, there are some variable. All the time you made a report about some practical lab, maybe it of chemistry, physics, and bio. There are some variables. The one is independent and the one is dependent variable. So there are some variables, or there may be some conditions such as light, water, carbon dioxide. These may be the condition, as well as temperature. These all is changed and a response of an other variable is measured. So in the experiment, one is independent variable while the other is dependent variable. Independent variables, the condition that is varied. Independent means it is free from everything. So the condition may be changed or can be varied timely. Dependent variable, it means is dependent on something. These conditions that response to the changes in the maybe something that can be provided to the experiment. So here is an example. To be a controlled experiment, it must have two identical groups. One is the experimental group while the other is control group. The group that is not exposed to the changes and experimental, because we are going to do experiment, so we have to change something in this experiment. So always experimental group maybe have some exposed conditions, while the control group always not always not be exposed with something and that can be changed. In for example, we consider an example of photosynthesis in which we provide carbon dioxide to the control group, while in experimental group we do not provide the carbon dioxide to the plant, because we have to draw some conclusions about the photosynthesis that we are doing in the experiment. So experimental group are not provided with the, and control groups are provided with the variables. So after doing experiment, what we do, make complete data. We do recording of the observation that we do. Okay, so what are the things that can be keep in mind about making a recording of the observations? That must be careful recording and must state how the experiment was planned, carried out, materials and equipments used, and how long it took. That is all the procedure that we do in a lab, and we should keep in mind that we have to keep in mind about the all the steps that we have performed. So because these all recordings that we made have to be used in the conclusion to draw results of the experiment of, or maybe last conclusions of the our problem about the scientific method. So what are differences between observations and inference? Observations and examinations of something in the nature detected by any of the five senses that may be our five senses: hearing, smell, taste, and touch. While the inferences are judgment based on your observations, it is a personal opinion that we say that all scientists have their own opinion about the observation that can that we may use for the hypothesis or not. So drawing and conclusions, this is the last step in making a uh, uh, solutions for the problem, and it always come at the end of the report. So what are drawing and conclusion? The answer to the scientific question is formulated by drawing a conclusion based on the data, scientific facts collected during the experiment. Statistics help form the conclusion. What are the statistics? Statistics are some the data in the form of numerical forms that we made in the tabular form, or maybe in the form of graphs that can be used to draw a conclusion. The answer to the scientific question is formulated by the drawing a conclusion based on the data. So the data help us to draw a scientific conclusions about the hypothesis or the observations. So after making a conclusion before accepting the conclusion, scientists retest their hypothesis several times. So this is not a single time process. The observation or hypothesis we deduct can be or may be retested again and again for the questions, because we said we saw that all scientists have their own opinion about the problem, so they retest their hypothesis again and again to check the validity of the solution. Later, the other scientists repeat the experiment until the hypothesis and the conclusions are spotted or rejected. If the hypothesis that a scientist is performing experiment on that show some doubt, then the hypotheses are rejected. Otherwise, if the experiment of the scientists spotting the hypothesis, they will appreciate it. Okay, so what comes after the conclusion? That is a terms that comes after making a repeated or skeptical approaches of the hypothesis that have been tested again and again. So what law and theory, when a hypothesis explain how the fact of nature occurs, it becomes a scientific principle or law. For example, as the law of gravity. So the law of gravity explain us about the attraction between the bodies. So it form a law because it is applicable in all the conditions under the gravity. So there is a simple hypothesis: if we move above the ground, the gravity becomes small. So this is a simple hypothesis and can be tested by a experiment, or it become a scientific law if it approve it. So when a hypothesis explain why natural events occur through the observation and the investigation over a long period of time, it becomes a theory. For example, theory of evolution or the cell theory, because if the virus if the virus considered as cell, then we can say that the theory of cell may be as scientific law, while the scientists do not include the virus as a virus in the category of cell. So that is the problem with the uh, that is problem with the variability of the cell theory. So all okay. So we can say that what theory is, if a hypothesis is tested again and again over several years or maybe at every time, and if it support the experiment or hypothesis, so then we say the hypothesis become theory. And if a theory support over a long time by experiment, then we say that the theory is a law, because theory explaining all the answers to the question that come from the different scientists. So scientific law, if a theory survive the skeptical approaches and continue to be spotted by experimental evidence, it becomes a scientific law, as we have heard Weinberg in the biology, in the genetics, and Mendel's law of inheritance also in the genetics field. These both are only, we have two laws in the biology, while the others only we have theory as a cell theory, theory of evolution, as an endosymbiote theory. These all may not support our questions, so we do not consider this scientific law, while we consider all these are theories of while all these are theory. So this is all about the scientific law. Now we will discuss a single experiment, and 1 minute. Okay, so now we will discuss a simple experiment that we can include in our today discussion, that is the study of malaria. So we will discuss here how we can solve a problem with the scientific method or with the help of biological method. So what is malaria? It is a disease that is common, what that was common in the countries including Pakistan, and we will go through the history of the disease to know the how biology solve the biological problem concerning the cause of the transmission of the malaria. Basically, the study of malaria is on the transmission of malaria from one places to another, and how physicians were familiar about the malaria more than for 2000 years ago. So we will discuss here. They described it a disease of chill. When the malaria was discovered, they called it as a disease of chill and fever with the recurring attacks, maybe a shivering of the body. They also observed, because the disease of chill, in this disease the body performs shivering without and without impact of the neurological approach, and it performs without and the actions that are not in our psychomotor control. So these diseases are of chill and we may feel cold, and they may be of recurring attacks. They also observed that the disease was more common among the people living in the low marshy areas. The people living in the muddy or marshy areas have more a problem with the disease. It was thought that the stagnant water that may be polluted water of the marshes poisoned the air, and as a result of breathing in the bad air, people got malaria. So the first observation that come in our mind that is the malaria may be caused by the breathing and that may be bad air. So the first observation they made about malaria disease, that is bad air. So they believed this belief led to the name of this disease. The Italian word mela means bad and area means air. For the further clarification of the observations, some volunteers drank stagnant water from the marshes. They did not develop the malaria. So check out there. The some observation that were made, breathing may be the cause of malaria. While when the volunteers drank the stagnant water, they did not got the malaria. So the next observation is disapproving the first observations. So there will be the need of the another observation. In the 17th century, when the New World America was discovered, many plants from the America were sent back to the Europe to be used as a medicine. That was quina. The bark of tree known as quina was used as a medicine to treat the malaria. Was very suitable for curing fevers. It was also beneficial that soon it was impossible to carry enough bark of the Europe, because they cannot cut more trees. So they thought that it was not beneficial to them. So some dishonest merchant began to substitute the bark of another tree, that was cinchona, which was closely resembled to the quina. So as they saw that the cutting of tree was not beneficial to them, so they substituted the bark of quina with the cinchona, which was similar to the quina plant. This dishonesty proved much valuable for the mankind, and cinchona bark was found as to be the excellent medicine for the treatment of malaria. We now know that the reason cinchona bark contain quina that is effective in treating the disease. So here come another observations that cinchona bark contain quinine that is effective in the treating of disease. So the first observation that was disapproved by the another, and another observation was made about the treatment of malaria, which was cinchona bark that contain quinine that is effective in treating the disease. So at that time, physician treated malaria with cinchona without understanding the cause of malaria. 200 years later, it was found that some diseases are caused by the tiny living organisms. So that is the era of Pasteur. After this discovery, it also became a belief that malaria too might be caused by the microorganisms. So this is another observation that was made. Which one is that it discovered that it also became a belief that malaria too might be caused by some organism. In 1878, a French army physician, Larvin, began to research for the cause of malaria. So he took a small amount of blood from a malarial patient and examined it under the microscope. He noticed some tiny living creatures. His discovery was not believed by the other scientists. 2 years later, another physician saw the same creature in the blood of the another malaria patient. 3 years after the second discovery, the same creature was observed for the third time. The organism was given the name as Plasmodium. So here we will make another observation that Plasmodium is the cause of malaria. The study started from this observation that malaria is caused by the breathing of bad air that was disapproved uh, by the drink of stagnant water by the volunteer, and they did not develop malaria. So another observation come that cinchona bark was a suitable or excellent treatment for the malaria because it containing a quinine in it. So after that, another observations comes that malaria is caused by the small organism, and this organism was given a name of Plasmodium. In the last part of the 19th century, many different causes of malaria were being studied. By the time, there were 4 major observations about malaria. These were the 4 observations that were made by on the malaria. Malaria and marshy area have some relations. So, as the first observation was made on the marshy areas, so they saw that malaria and marshy area have some relation in between them. And after another observation was made, quinine is an effective drug for the treatment of malaria. And after that, they made another observation: drinking the water of marshes does not cause malaria. So, and at the last, plasmodium is seen in the blood of malarial patient. These were the 4 observations that were made from 1878 to 19th century. Okay, we know that the scientist use whatever information and observation he has and make one or more hypotheses. So we told about that hypothesis always made from the observation, and observation can be of 2 types, quantitative and qualitative, and all the hypotheses that are made must be answerable to the questions. So here we are with the last and the recent observation, that is plasmodium is the cause of malaria. So the scientists took this as a hypothesis, the plasmodium is the cause of malaria. So they, after getting this hypothesis, they check it how scientists does not know whether this hypothesis is true or not. As we told that, or we discussed that the hypothesis may be true or may not be. So scientists do not know that the hypothesis is true or not, but he accept it may be true and he make deduction. We made deduction only when we accept it as a true hypothesis. Otherwise, the hypothesis is rejected. One of the deduction from the above hypothesis was made. So it also starting with the if and proceeded with the then. If plasmodium is the cause of malaria, then all the person ill with the malaria should have plasmodium in their blood. So this is our deduction from the hypothesis that we made from, and this is if plasmodium is the cause of malaria, that is our observation. Here plasmodium is seen in the blood of a patient. So if plasmodium is the cause of malaria, then all the person ill with the malaria should have plasmodium in their blood. That is the result. Okay, we have to check this if this is true. The next step was to test the deduction through the experiment, which were designed as, so after making a deduction, we have to perform an experiment to check the feasibility of the problem or the hypothesis we have made. So how they did the experiment? Blood of 100 malarial patient was examined under the microscope. So check out this. This is the quantitative or qualitative observation. We are getting some numerical value here. So these are about some numerical values. So we may get some numerical approach to get the consequences of the experiment. So this is the quantitative observations. Blood of the 100 malarial patient was examined under the microscope for the purpose of having a control group. So we also discussed that the experiment is made through the two groups. The one is control group and the other is experimental group. So for the purpose of having a control group, the blood of 100 healthy person was also examined under the microscope. There were two types of person, one that were healthy and the one that were uh, uh, ill by the malaria. So the patient that do not have plasmodium in their blood, that were called as a control group, while the patient that have malaria that have plasmodium in their group was called as a experimental group. So blood of 100 malarial patient was examined under the microscope, and for this purpose they made a control group. The result of experiment showed that almost all the malarial patients had plasmodium in their blood, while the 7 out of 100 healthy person also had plasmodium in their blood. Now we know that the plasmodium in the blood of the healthy people was in incubation period, i.e., for example, the period between the entry of parasite into the host and appear as a symptom. The results were quite convincing and proved by the hypothesis plasmodium is the cause of malaria. So this is our hypothesis, plasmodium is the cause of malaria, and we did an experiment on the 100 people. Some of them were ill by the malaria and some of them were healthy persons. So the result showing that only 93 people had plasmodium in their blood, while the 7% from the healthy person were also had plasmodium in their blood. That means the plasmodium is in their incubation period. What is incubation period? A period in which parasite enter the host, but it do not show the symptom, while it keep the process of harming the organism. So this show that the results were convincing and support our hypothesis, because we have seen plasmodium in the blood of a malarial patient and in the healthy persons also. So malaria has killed more people than any other disease. The account of malaria is an example of biological problem and of how such problem were solved. Next, biological problem was to learn about how plasmodium get into the blood of man. So this is the another approach to another observation, how malaria transmit from one person to the another person. So biologists were having following observations. The one is malaria is associated with the marshes, that we said in the 4 observations that we made here, that malaria and marshy area have some relations. And the other one is drinking water of marshes does not cause the malaria. These both were the previous observation, and we always says that the observation that we made are always the previous data that have been performed by the our pioneers. So the scientists that made experiment or the research on how plasmodium get into the blood of man were considering the observation that already made by the other scientists. So these were the two observations that were made, and now we will see how these observations were performed by the next scientist. From these observations, it can be concluded that plasmodium was not into the marsh water. So these two observations showing that the plasmodium that is transmitting from one person to another is not present in the marshy or stagnant waters, but it must be carried by something that comes from that comes to marsh water. Means there is a transmission medium that can transfer the plasmodium from one place to the other. And in 1883, a physician A.F.A. King listed 20 observations. Some important observation of the king were these are the some observation by the A.F.A. King. People who slept outdoor were more likely to get malaria than those who slept indoor. That were the observation made by the A.F.K. King. Those people slept outdoor without closing their door to the marshy areas may get malaria in their plasmodium get in their blood. People who slept under the fine net were less likely to get malaria than those who did not use such nets. This is the same cause as dengue, or that is our recent problem, and it may be about the mosquitoes. So people who slept under the fine nets were less likely to get malaria than those who do not use such nets. Individuals who slept near the smoky fire usually did not get malaria. So this is maybe the solution of something, and that is caused by the malaria, showing that a smoky fire uh, exhausted or con क्या कहते हैं individual who slept near the smoky area were did not get malaria. On the basis of this observation, King suggested a hypothesis. The one is mosquito transmit plasmodium and so are involved in the spread of malaria. So the king suggested mosquitoes transmit plasmodium and are also involved in the spread of malaria. This is the first observation that were made by the king from these two observations. Following the deduction were made, this is the hypothesis that were made from these observations that king made. Okay, so following the deduction were made considering the hypothesis as true if mosquito. This is our deduction. If the mosquito are involved in the spread of malaria, then this is our deduction or our observation, and this is our this will be our conclusion or our experiment if this come true. If mosquitoes are involved in the spread of malaria, then plasmodium should be present in the mosquitoes, and a mosquito can get plasmodium by biting a malarial patient. So in order to test the above deduction, Ronald Ross, a British army physician working in the India in 1980s, performed important experiment. He allowed a female Anopheles mosquito to bite a malarial patient. What he did only, he just allowed the female Anopheles mosquito to bite a malarial patient. He killed the mosquito some days later and found Plasmodium multiplying in the mosquito stomach. When the Anopheles bited the malarial patient, he killed the mosquito some days later, and the Plasmodium multiplied in the stomach. So the one thing you all may know, the only female Anopheles can cause the or may cause the transmission of Plasmodium into the next generation, while the male do not have this ability. So the next logical experiment was to allow an infected mosquito having Plasmodium to beat a healthy person. After this experiment, what he did, he started experiment of allowing an infected mosquito to bite a healthy person to saw that if the person is getting the Plasmodium or not. If the hypothesis was true, the healthy person should have got malaria. This is our hypothesis. But scientists avoid using a human being for the experiment. This is the approach by the scientists do not use the human as a experimental group for any problem related to the humans. So what they did, they just uh, get a sparrow and allow a Culex mosquito to bite a sparrow for the experiment. Some of the mosquito were killed and studied at various times. Rose found that Plasmodium multiplied in the walls of mosquito stomach and then moved into the mosquito saliva gland. So these all are the experiment were performed by the Ronaldo, and they concluded that the Culex mosquito that have bite the infected person, and after that he bited a sparrow. The both were carried Plasmodium in their blood, and that is multiplied in the stomach of sparrow, healthy sparrow. So these all are the tests and the experiments that can be used to perform an biological or scientific problem to solve any biological or life problem. We can use a scientific method, or all the time you made a report about any subject, you have to follow this all step. And all the time if you going to made a research on something, you have to perform all this process inductively or inductively. These were mentioned here. The first one is you have to define a problem. First, you have to get some observations about the problem. Then you have to collect the background information about the problem. After that, you made a hypothesis. Then you made experiment. Before experiment, you made a deduction. If the hypothesis were spotted by the experiment, then we make a record and the next observation and draw conclusion. And if the conclusion of this all procedure again and again spotted by the test is, so this become as theory. And if theory spot all the answers that scientists can ask or may induce about the problem, they they made, so that automatically converted into the scientific law. So these all are about the scientific problem that we can use in our daily life also, or in the biological field as well. If you all have any questions about the scientific problem, or about anything else, you can ask me. Dog rabies. Yeah, it is also a disease that is caused by the bacteria also, and it may be when we studied the virus, that also caused by the some strain of the viruses, and we discussed there, maybe I guess we discussed. No. Okay, if you have any question, you can ask me.
Guys, is clear, or do you have any question? Remember, if you have some question, just turn your microphone and put at the mute and ask the question to the teacher. He's going to answer you, okay? Is clear, guys? Yes? Yeah, the guy say yes, everything clear. So actually, Danny say everything is clear, but what happened with the other guys? What happened with the rest? Is clear, guys? Camila, Alejandro, what happened? Is clear, guys? Okay, you can continue, teacher. You can continue with the explanation, okay? It's 1 hour the class. Let me know when it's done, okay? When you complete the class, okay? Okay, Mohammed?
Yeah, class is complete now.
Complete, okay, excellent. Very good, teacher, excellent class. Guys, thank you for being here. And remember, be pending for next class today, 3 p.m. It's the Spanish class, okay? So enjoy the day, guys, and thank you so much, Mohammed.
Okay, thank you.
Very good. The class is done, guys. Have a wonderful day to all of you. Wonderful day, Mohammed.
Okay, thank you. Okay.
Bye.
Bye.
Bye.